actually have an aunt with an autoimmune disease, and I was just wondering, do you think that this immunotherapy that you're talking about, this immune system, would that work for her situation, or is this only relevant for cancer patients?
The role of the immune system is important in healthy tissue all the way through all kinds of diseases, but we can't really talk about, I don't know anything about your aunt's personal situation, and that's well beyond the scope, I think, of what anybody in this group would know.
Oh, so you couldn't actually recommend something for her. You wouldn't be able to do that.
No, definitely not. Yeah,
Okay,
she would
thanks
need
for
to talk to
clarifying.
her physician.
Got it. Thank
And
you.
that's a really important question, Christine. I just want to remind everyone that both Oli and Aaron are PhD researchers and they are not clinicians. So please, we try not to ever ask any personal medical questions, whether it's about you or someone you know. If you can reframe it in a general question, that would be great. But unfortunately, they cannot give us personal medical advice.
Right. So continuing on from there, um, right, in order to have immunotherapy work, you need different things to be presented on the cell surface. And specifically for cancer immunotherapy, targeting CTLA-4 or PD-1, and then the PD-1 ligand to remove the inhibition of those T cells would improve that anti-tumor response, right? And how those things are localized to the surface and how they get there, how they stay there, etc., right, is something that is still being tackled. So in previous work, we've shown that
It
uh,
it looks like Bob might be a little confused.
Oh, well, thank you. I was just thinking, are those checkpoint inhibitors? I mean, that's a term I know, and I see the PD-1, PD-L1. I think that's a checkpoint inhibitor, but I just want to double check that.
Yeah, Oli, could you maybe contextualize that a little bit for them, or I can if you'd rather.
um, yeah, I think I might need to check my notes if you've got a ready answer, Erin.
Yeah, sure, happy to help. So yes, CTLA-4 and the PD-1, PD-L1, those are two proteins. One is on the cancer cell, one is on the T cell, but they bind to each other. So it's the two sides of that lock and key that Oli was talking about earlier. Those are both sort of the pathways that are clinically targeted by pembrolizumab, and you all actually probably know the clinical names better, Keytruda, but all of the immune checkpoint therapies are what target the CTLA-4 and the PD-1. Yeah.
Thank you.
Did that answer your question, Bob?
It did. It was very helpful. Thanks.
Okay. And Maggie and Michelle, how are we doing? Do you have any questions at this point? You're good. All right.
Okay. So RAB11B is a protein that was found to help mediate endosomal trafficking, which just means that it's going to regulate what's coming from the cell out onto the surface, which is obviously key to presenting those keys for the locks there. And so we investigated kind of the role of that trafficking and found that there were some family interacting proteins, which I'll call FIPs from now on, specifically RAB11, FIP2, and FIP3, that are required for breast cancer cells to be recognized by CD8 positive T cells, specifically in the context of a really like immunogenic compound being presented, in this case of albumin. And our model for that is this infecal model, that is the peptide that's being presented on those cells. But when we did flow cytometry, we didn't see that it was due to a decrease of presentation of those keys, the PDL1 or this infecal complex on the MHC. So that's
Could
kind
I
of
interrupt for just a second, Oli? Bob,
yeah.
it looks like you might be a little bit confused about something. I'm just trying to read your face. Do you have a question?
Well, not really a specific question, but this is just a lot of science. I'm just trying to follow it because there's a lot of terminology and a lot of things I don't really understand. So I'm just trying to catch up.
Was there
Maybe
something specific, some terminology that you would like explained?
No, it's almost the I was just trying to catch up with what kind of the big picture of where we are.
Okay. All right.
Yeah.
Actually, I think maybe if I go to my figures, it might be easier to follow along with this if I have it illustrated.
Yeah, and if you
Sure.
can just kind of zoom out and put everything more in context of really the disease instead of maybe worrying about going through and explaining every Western blot and every control in your flow cytometry. Yeah.